Hi, my name is Josh Meyer, and I'm honored to be delivering this talk at this conference entitled Genomic Differentiation of Early Onset Colorectal Cancer. We're going to start off talking a little bit about the incidence of early onset colorectal cancer, and then we're going to move into the natural history, looking particularly at the lymph node involvement of this particular disease. We'll then talk about the mutational landscape of early onset colorectal cancer, and then spend a little bit of time discussing specific mutations in a gene called KRAS. So when I was a resident, I was in clinic and was looking around and seemed to think that I was seeing more rectal cancer patients than we would typically expect who are young, who are under the age of 40. And so we started looking into this and actually found that there was an increasing incidence of these younger patients with rectal cancer and actually with colon cancer as well. So you can see here that the colon cancer graph is on the top. And if you start looking in the mid-1980s, you can see the incidence is rising through the years up till about 2005. If you look at the rectal cancer graph below, you can see that it is rising even more steeply. When we looked at the rectal cancer data and we tried to identify when this rise began, we pinpointed it down to the year 1984. Where the slope started increasing. And you can actually see during this time that the annual percent change of rectal cancer in patients under 40 was almost 4%. And since we published this, and a number of other groups have published similar papers, and you can see here, this is a recent one where they analyzed patients by the year that they were born, otherwise known as the birth cohort. And if you start in 1950, you can see that colon cancer is increasing in incidence and rectal cancer is increasing in incidence as well, all the way up through people born in the year 1990. Here, I'm showing you the age distribution of patients with colon cancer, and it's divided up by T stage. So we have the T1 tumors on the upper left, and then T4 tumors down in the lower right corner. And what you can see is that there is a sizable percentage of patients who are young, who are diagnosed with this cancer in all stages. You can also see that there is a particular uptick in the diagnosis, especially in T1 and T2 stages at the age of 50, which has been the recommended age to begin colorectal cancer screening for average risk adults. As you probably know, in 2018, the American Cancer Society was the first major organization to call for a lowering of that screening age. And that was due to research like our group's research that has described this increasing incidence. And this is the headline from the New York Times in 2018. And then just on October 27th, you can see that the USPSTF, the biggest organization responsible for screening guidelines, came out with a recommendation to do the same thing, to drop the age of first screening to 45. And you can see a nice quote here from Michael Sapienza talking about how important a step this is. So we wanted to know a little bit more about the biology of these cancers. And the first thing we did was to look at these cancers and try to figure out if they were spreading to the lymph nodes in the same way that older patients' cancers do. And so this is a graph from our publication where we separated these cancers out by the T stage. So we have T1 on the left and T4 on the right, and then also by the number of lymph nodes examined, because if you examine more lymph nodes, you're more likely to have a lymph node that is positive. And you can see these are graphs that have the darkest blue bars with the youngest patients, and as they get lighter, those are the older patients. What you see is in every single group, the youngest patients have the highest risk of a lymph node that is positive. This is another figure from our publication where we looked at the cause-specific survival, or the likelihood that the patients were not dying of rectal cancer, based on the number of lymph nodes that were positive. And we did this 4 different times for different ages of patients, with the youngest patients on the upper left, age 20 to 39. And what you can see here is different graphs, and the colors denote different numbers of lymph nodes positive, with blue being 0 lymph nodes positive, and the gray being 7 or more lymph nodes positive. And you can see in every age group, your survival goes down as the number of lymph nodes goes up. So it's not just that younger patients have a more vigorous immune system and you're finding these lymph nodes positive, but those are actually meaningful lymph nodes, and it makes a difference. If we look at patients with early onset colorectal cancer, what we find is that it's a minority of patients who are being diagnosed with this because they have a hereditary condition. It's approximately 14% or so that's hereditary. The majority is actually sporadic, meaning there is no family history of cancer. And then there's another 15% or so that have either a first-degree relative with colorectal cancer or a first-degree relative with an advanced adenoma. We're going to talk a little bit about mutations, so I just want to remind you what a mutation is. So if you look at this figure and see that you have a normal strand of DNA on the top, where they describe it as the original sequence, and there are different bases that are aligned in a particular sequence there. And then if one of those bases changes, so in this example, from a T to a C on the bottom, that would be a certain kind of a mutation, just meaning a change in the DNA sequence in that particular gene. So we wanted to know whether there were more changes in certain genes, in one gene or another, in colorectal cancer among young patients as opposed to older patients. And so this is a study that I did with Chris Liu from University of Colorado, who's also speaking at this conference. And what we examined were a variety of genes known to be important in colorectal cancer. And we found that actually there are differences. So if you just look at younger versus older, you can see that there are differences in these genes that are known to be important, like TP53, APC, KRAS. These are all genes known to be important in colorectal cancer progression. And we did find that they are differentially mutated in younger versus older patients. Sometimes more, sometimes less. If we look at the genes that have an increasing mutation as the patients get older, here we weren't just looking at 2 categories of young versus old, but actually all of the patients are included together. And what we see is there are a number of genes where as patients get older, they have a higher risk of that mutation. So here we looked at 10 genes, but then if you zoom in, you can see there were 2 in particular that were pretty interesting, both important genes for colorectal cancer, one called BRAF on the left and another called RNF43 on the right. And you can see as the age of the patient increases, the risk of a mutation goes up. When we looked at these a little more closely, we found something very interesting, which is that it's not just any mutation on the gene. It's not that it's random, but actually it's a very specific mutation that's driving this increase. So on the left with BRAF, there's a particular mutation where a single spot is mutated from one to another codon, one specific spot where there's a mutation. And that accounts for all of the increase as patients get older. And if you look at all of the rest of the mutations on that gene, they're flat. They're not hardly increasing at all. And the same thing is true for RNF43. It's a very specific mutation that's going up as these patients get older. There were also a number of genes, important genes, that were decreasing as patients got older. So this is not random. This is very specific. And some of these, again, are important genes for the development and progression of colorectal cancer. We then focused on a gene called KRAS. This is a gene that's chronically activated in about 30% of human cancers, and it's actually responsible for 1 million deaths per year worldwide because of that. This is a powerful switch that controls multiple cellular processes, and about 42% of colon cancers have a mutated KRAS, which is second only to pancreatic cancer in terms of frequency. You can see on the right is actually a diagram that we were able to construct, which plotted out all of the different mutations that we found. And you can see the red is the most common site of mutations. And although these mutations in the gene are located far apart from each other, when the protein is formed and folded up together into its structure, they really coalesce in that red spot in the middle. So the question is, does it really matter? I'll tell you all these things about, a specific mutation here versus over there. Why should we really care? Well, it does matter. And this is an example of why. This is a nice study where they looked at where is the mutation in either KRAS, a related gene called NRAS, or another related gene called BRAF. So you can see here, the white side of this is the patients with a wild type or normal copy of these genes. And on the left side, you can see all of the different spots where patients have a mutation and what those mutations are. And they plotted out survival or disease-specific survival. In other words, the likelihood of not dying from colorectal cancer in this plot. And if you look at the green graph, that is all of the patients with the normal or wild type genes. If you look at the blue graph, those are the patients with G12 or G13, a specific spot in that gene mutation. And those patients clearly do worse, do not survive as long. But if you look at the red graph, those are patients who also have a mutation, but those patients did incredibly well. Their survival was really 100% in this graph. So it's really interesting. And the other reason it's important is because we now have drugs that are targeting these specific mutations. So this is an example of a study that was published just recently in the New England Journal of Medicine with a drug that works just on patients with a KRAS mutation called G12C, a very specific mutation. And this happens to be lung cancer, but there were also colorectal cancer patients in this study who responded. And you can see there were a lot of patients who responded to this drug, and we've never really had KRAS targeted drugs before. So this is an exciting breakthrough for us. So we looked specifically at KRAS mutations by age. And you can see here that we plotted these based upon exactly where the mutation is in KRAS, either for younger patients or older patients. And on top, you can see the younger patients had more mutations in this spot called G12 and fewer mutations in the green band or the orange band of A146 or Q61. So when we looked a little further at G12, we also found that the specific mutation, what we're changing the G to, is different for younger patients versus older patients. So we talked about why that matters. There are specific drugs and there are specific prognostic features associated with the particular mutation there. And so these are different. They're not exactly the same. We also looked at this Q61 position, and interestingly here, we found something that's different as well. Particularly interesting was this dark blue band, the Q61K, that we found in the older patients. So this is a newly described mutation that no one had found before, that we really only found in older women with colon and not rectal cancer. So older, female, and colon. And it's really interesting. It just tells you that there are small subpopulations here who are going to behave differently, and we should expect to respond differently to treatment. So in conclusion, colorectal cancer incidence is certainly rising among younger patients, and the organizations associated with screening guidelines have really begun to come around and take note of that. The etiology is not clear, but the data suggests that these younger patients may in fact have a distinct biology, and there are numerous genetic differences in colorectal cancers of younger patients that we've been able to find. And with some additional effort, these differences in genetics may lead to changes in the ways that we approach these patients in the clinic. I'd like to thank, of course, the AllyCon convention for asking me to give this talk, and I've had a lot of help with a lot of these studies that I was able to present to you, both inside Fox Chase and then outside Fox Chase Cancer Center, where I work, as you can see here. And then, of course, I truly appreciate the funding from the Colorectal Cancer Alliance and the CRISPR for Life mechanism that has really helped me to be able to do this work and help the whole team to be able to do this work. So I very much appreciate that. Thank you for your attention.